We are going to start today with Silvia Pittolo, who is currently in University of California in San Francisco, where she is fortunate enough to do a postdoc, and she'll be moving back, I believe, actually to Europe in not too far a future. But what she's going to do today is talk to us a bit about dopaminergic activation of prefrontal astrocytes, requires both dopamine and adrenergic receptors. So Silvia, if you please.
Thanks, Ulrik, for the introduction, and I'll share my screen. Hopefully you'll be able to see this. Let me know if you don't see it. Okay, so thanks for the introduction, and I wanted to thank the organizers for creating such an inclusive conference. And I'll start right away by explaining why we're interested in studying astrocytes and dopamine. Dopamine is a monoamine that is crucial to many brain functions, such as control of movement, learning of conditioned behaviors, but it's also involved in disorders such as Parkinson's, drug abuse, depression. And dopamine, like other monoamines, is produced by few subcortical nuclei that have huge axonal arborizations. And so despite their size, they can really broadcast signals to basically the entire brain. And other monoamines project to the entire cortex, but cortical inputs for dopamine are mostly limited to the prefrontal cortex, which is an association area and a candidate for decision making. And dopamine is also a slow neuromodulator as opposed to fast neurotransmitter like glutamate and GABA. And this is in part due to dopamine receptors, both the D1 and D2 subtypes, being exclusively G protein coupled. And so dopamine works on relatively long timescales. And in this context, astrocytes seem the perfect candidates for listening to a broadcast signal such as that of dopamine. First, because astrocytes in one of their territories can encompass up to thousands of synapses, and then neighboring astrocytes can connect through gap junctions and form a network that can really sense the activity of large populations of neurons. And another reason is that astrocytes mostly express G protein kappa receptors, and they've been shown to work on long timescales compared to neurons. But really what we know about astrocytes in the PFC, the prefrontal cortex, is very limited. And today I'm going to show you work that we did to address the question of whether astrocytes in the PFC can sense dopamine. But first, I'm going to talk about how the activity of prefrontal astrocytes looks like in vivo. And this may seem trivial, but the prefrontal cortex is very deep, so it needs to be accessed using microendoscopes, which basically are lenses implanted in the brain. And then the animal can be head fixed and imaged through a conventional 2-photon microscope. So Sai Yokoyama, a research technician in the lab, used this method to image mice whose astrocytes were expressing a fluorescent calcium sensor, like GCAMP. And to the best of our knowledge, these are the first images of prefrontal astrocytes in vivo. And she saw something quite interesting, especially comparing to a sensory cortex like primary visual cortex. And so in V1 work from our lab as well as others has shown that astrocytes can become active in bursts of activity that span across the entire population. And here I'm showing an example of this type of activity, and this was done by Michael, a grad student in the lab. And you can see some sparkles of calcium and then this type of coordinated activity or bursts. And I want to point out that our understanding of astrocyte activity is still limited because we're still developing the computational tools to analyze such a complex activity. And so here, I'll play it again. On the top, I'm showing the raw fluorescence video, and on the bottom, the calcium activity as detected using AQUA, which is a software that our lab developed and was published last year. And once I looked at astrocytes in the PFC instead, I'll play this video, she didn't see that kind of activity, but she saw rather discrete calcium events like these. And this is here just a reference for the size of individual astrocytes, so you get the sense. And so we quantified a few animals, and we kept seeing that in the PFC, astrocyte calcium events couldn't get as big as in the visual cortex. And this was really intriguing because it suggested that astrocytes are not a homogeneous population across the brain, maybe have different roles in different circuits. And we wondered whether these events we see in PFC in astrocytes could be driven by dopamine. But to understand this, we first needed to answer whether dopamine receptors are expressed on PFC astrocytes. And this seems trivial also, but other groups have shown D2 expression only by antibody detection. And we wanted to move away from antibodies against dopamine receptors because their specificity is low. And so we decided to take a different approach. And so Psi crossed transgenic reporter lines for astrocytes, so expressing fluorescent proteins in astrocytes, with reporter lines for expression of D1 or D2 receptors. And Psi found colocalization of astrocyte markers indicated by the white arrows with markers of both D1 receptors and also D2 receptors. And so we found that prefrontal astrocytes do have dopamine receptors. Then we wanted to know what signaling pathways were activated downstream. And we first decided to look at cyclic AMP, as D1 and D2 receptors are canonically linked to GS and GI, and respectively activate and inhibit adrenergic cyclase. And the way we approached this was by injecting mouse pups with an adeno-associated virus that encoded for a cyclic AMP sensor called primfamindo under an astrocyte promoter, GFAP. And 4 weeks later, we sliced their brains and we imaged them under a 2-photon microscope. And we used a cocktail of drugs to block neuronal activity because we really wanted to isolate pure astrocyte responses. And then we buzzed on specific agonists for D1 and D2 receptors, then dopamine itself, and finally an adrenergic cyclase activator, which is for scoring, as a positive control. And this is an example of how the astrocytes look like in a prefrontal cortex slice. And this is a kind of experiment we did. And so we first applied the D1 agonists, and we didn't see an increase in cyclic AMP, which is what we were expecting. But we did see an increase with a positive control for scoring, meaning that the cyclic AMP sensor was working. And this is a quantification, and the dots indicate slices. And the same thing happened with the D2 agonist. We didn't see any recruitment of the cyclic AMP pathway. And finally, with dopamine, the same thing happened, no response. And so it seems like dopamine does not mobilize cyclic AMP in prefrontal astrocytes. But because others have found that dopamine receptors in neurons can function in non-canonical ways and release finally intracellular calcium, we checked this pathway too. And so we targeted astrocytes again by injecting a virus, this time encoding for a calcium sensor, GCAMP, and we again blocked neuronal activation to observe astrocyte responses. And then we both applied dopamine and dopamine receptor agonists. This is an example of an experiment in slices. On the top, you see the raw fluorescence, and here the aqua detection of the calcium events. And you see that after dopamine is applied, astrocyte calcium is recruited. And so with aqua, we can plot this as time course of the calcium events and position on the y-axis, and the colored line indicates when dopamine comes in. And then we can extract the area and the duration of all these events, and we see that both these parameters, both area and duration, increase after dopamine. But if we swap dopamine with a specific agonist for D1 and D2, we are unable to reproduce this response. And so we found that dopamine mobilizes calcium, but if we activate dopamine receptors directly with specific agonists, this does not happen anymore. And so we wondered which receptors were actually responsible for this calcium mobilization downstream of dopamine only. And to tackle this, we turned to another approach. Instead of bath application, we use the caged form of dopamine in the bath, and we release dopamine by light stimulation, which allows fast release that can be repeated over and over on the same cells. We again block the neurons, and we did this with caged compounds alone, or while blocking dopamine receptors to see their contribution. This is again an example. Oh, this is not playing, sorry. I can pull it up from here if you don't mind. So this is an example of the uncaging. You see 3 white stripes that correspond to the uncaging. And this happens here, dopamine is released, and then you see astrocytes being activated. Sorry. And hopefully I can get back to this. And so if we quantify the response of these cells in the control, we see no difference in the number of calcium events between before and after the uncaging. Oh, now it works. So the uncaging is indicated by the blue box. But in the presence of the caged compound, the caged dopamine, after uncaging, we see an increased activity. However, when we block the dopamine receptors, both D1 and D2, the response in the same cells is reduced but is still present, meaning that the dopamine receptors are not the only target for dopamine. And from other experiments that we did, I don't have time to show them, we knew that the response was due to a GPCR, and so we thought that maybe adrenergic receptors could be recruited because of the similarity between dopamine and norepinephrine. And so we explored this possibility by blocking adrenergic receptors, both either alone or in combination with the dopamine receptor antagonists. And this time we saw a suppression of the response, suggesting that adrenergic receptors are required for astrocytes to respond to dopamine. And finally, what were the dynamics of the response to dopamine? We see a response in less than 5 seconds after the uncaging, although this takes longer when we block the dopamine receptors. But there is no response with adrenergic receptor antagonists. And finally, the duration is about 15 seconds. And so how do we explain these results? What the literature says is that if dopamine receptors were expressed on astrocytes, they would activate cyclic AMP. Instead, we see calcium. We also know that the response depends on adrenergic receptors, and in this case, the literature says that the alpha-1 subtype of adrenergic receptors can recruit calcium. And so our current hypothesis is that dopamine can bind both to dopamine and adrenergic receptors, and that this interaction mobilizes calcium. In fact, if we only activate dopamine receptors with selective agonists, we don't see any calcium mobilization. When we block the dopamine receptors and apply dopamine, the response is dampened but still present. And finally, when we block adrenoceptors, which are likely the permissive subunit for the calcium signaling, the signaling is blocked. And so why does this matter? We think our findings are important in many ways, but first of all, we're showing that astrocytes in the PFC are integrating dopamine signals. And so we think that in future studies that want to decipher the actions of dopamine in the PFC, we should take into account astrocytes too. And also adrenergic receptors seem to be necessary for a proper response to dopamine. And so we think this opens the discussion on interactions between dopamine and the adrenergic system and how this might be important for treating psychiatric conditions. And with that, I'd like to thank you for listening. And I'd also like to thank all the people in the post-cancer lab, but especially Kira, who was a great mentor, Sai, who did all the green lens work in PFC, Michael, who imaged in visual cortex, Michelle, who gave me great advice all the time, our collaborators at Virginia Tech for the aqua development, and our funding sources. Kira is funded by the NIH and NSF, and I am funded by an individual fellowship from the European Union. And with that, I'd like to take any questions.
Thank you. Excellent. Thank you, Silvia. And perfect timing too. Well done. That's very cool stuff. A lot of it is outside of my realm. But if I can start off with a question first, maybe. I'm quite interested in the role of dopamine and how it relates to things like motivation, how it relates to things like learning to, and other kind of behavioral measures. And you kind of touched a little bit upon, how that is something that this kind of interaction between the astrocytes and dopamine might actually have some kind of effects on. And can you kind of get it down to a low enough level that I might be able to kind of follow some of it too?
Yeah, so I mean, this is a very interesting question, and we would love to be able to answer this, but there are so many things that we still don't know about how the astrocytes talk to one another and talk to the neurons, and so it's really hard to to predict what would be the implication of astrocytes in these motivational and learning circuits. And we haven't done any experiment to tackle that. So.
Sure. But I mean, it's certainly true that it's you tend to not, those of us who do any kind of modeling of dopamine, we don't tend to think about astrocytes at all.
Mm-hmm. Yeah, right. So. You should add them into your models. It would be really interesting. I know that there are a few models of how astrocytes actually work. And I think in the computation of the circuits, they are, I think, really important because astrocytes can hear or listen to neuron activity. They express receptors for GABA, for glutamate, and so they can listen to neuronal activity, but also they can modify. And I think maybe later on we'll hear other people talking about that. But the astrocytes can actually modify the functioning of the circuit. And so, for example, they can uptake glutamate, and they can also release ATP, which also activates neurons to some extent. Especially, it changes the excitability, right, of the circuit. And so, I think they are important in the functioning of the circuit. And something that is really interesting, and I wanted to explore. I don't know if I will have time before moving back to Europe. I wanted to check whether this big calcium activity in the astrocytes actually leads to some kind of change in the potassium balance, because astrocytes are very important for balancing potassium. When there is a lot of neuronal activity, they take up excess potassium. Okay. And so I'd like to look at that. I don't know. Yeah. We'll see.
Plenty of unanswered questions, as always. There is a question in the chat. Unfortunately, we don't really have time to look at that right now. So I hope hopefully that Silvia and the person asking the question can have a chance to look at it afterwards.
Thank you.
Thank you, Silvia. So if you can unshare, stop sharing.